Hi, everyone. Thank you so much for joining our webinar today titled Let It Flow, Flow Cytometry and Probiotics, Q&A with the experts. I'm very excited to have Dave and Anjay here with me today. I'm Amy Knock, and I will be your host for today's session. Before we begin, I just wanted to let you know a little bit more about how the webinar will run. The webinar is being recorded, and the recording will be available to you within 3 business days. I just want to advance the slide here to give you a brief update on how the webinar will run today. So the format of today's webinar is comprised of questions submitted from the Let It Flow webinar presentation that we had earlier. During the webinar today, you can submit questions you have using your webinar sidebar menu. Select the Questions tab, type in your question, then hit the Enter key on your keyboard. And remember, you can submit questions anytime throughout the webinar. We will be reviewing these and responding to them during the presentation. If we don't have time to address them during the live session, a Eurofins rep will follow up with you after today's webinar. All right, that concludes my brief introduction. Dave and Anjay, I will now pass it over to you. You may begin the discussion.
Hi, everyone, and nice to be speaking with you again. In case you weren't on the call last time, my name is Dave Roth. I'm Business Development and Client Service Director for Eurofins, supporting our probiotic business. And I'll be acting as the moderator today to ask the real expert, Anjay, all the questions today. So first, we thought it would be a good idea to just start with a little bit of a refresh regarding flow cytometry as a technology, specifically for probiotic enumeration. So Anjay, could you give us just a little bit of a refresh on how flow cytometry is used to enumerate probiotics and what the technology is?
Sure, yeah, thanks, Dave, and thanks everyone for joining this morning. As Dave and Amy said, my name is Anjay Benkowski. I'm the technical manager for probiotics and dietary supplements with Eurofins based out of Madison, Wisconsin. And today, as we mentioned, we're going to be answering some of the questions that we received during our last webinar that was presented a few weeks ago in regards to testing probiotics by flow cytometry. We've been testing probiotics and enumerating probiotic materials by flow cytometry since 2017, and we are following ISO protocol, ISO 19344 protocol B, and what this is measuring and what we're using flow cytometry for is to measure cellular viability based on its membrane integrity. So what we do is we take the samples and we stain them with dual nucleic acid stains. One stain is able to penetrate cell membranes, and that's the live stain. And then a dead stain is only able to penetrate compromised cellular membranes. And then as the cells pass through the interrogation point on the instrument, they're excited by a laser system, and that emission is then turned into a voltage pulse on a photomultiplier detector. And that shows up on a dot plot, and we get discrimination based on the different emission spectra of the live and dead cells. So what we end up being able to see is a live cell count expressed in active fluorescing units, injured and dead cell counts as well, and those are expressed as non-active fluorescing units. The entire cellular population as a whole is considered the total fluorescing units, and we found over the years that there's a number of advantages that we've seen with flow cytometry, including real-time result generation. The gold standard for probiotic enumeration, as many of you know, is the cultural plate count, and that typically takes anywhere from 2 to 5 days of incubation to allow colonies to grow on the plate, whereas flow cytometry can produce enumeration results in real time as it's being run on the instrument. We've also shown in our research that it has improved precision over plate count assays, and of course it's a different measure of viability comparatively. So plate count enumerations are measuring cellular viability based on the cell's ability to replicate on a Petri plate or ideal growth conditions, whereas flow cytometry, as I mentioned, is measuring viability based on cellular membrane integrity. So I think with that level set, we can hop right into the questions, Dave, if you'd like. And for those that are on the call, please feel free to enter your questions in the chat, and hopefully we can get to as many as we can in this session.
Great. So we do have a pretty good list of questions from last time's webinar. So let's jump right into those. So the first one is, can you use flow cytometry for yeast enumeration? And can you differentiate yeast from regular bacteria when using flow cytometry?
Short answer is yes. We have found that we've been able to apply the ISO 19344 protocol B using the same nucleic acid stains as prescribed for lactic acid bacteria to evaluate probiotic yeast such as Saccharomyces cerevisiae, subspecies Boulardii. It stains in a very similar fashion. So we're able to use the same parameters that we use for lactic acid bacteria. We do have the ability to distinguish based on cell size, which is one of the core components of flow cytometry, looking at forward scatter and side scatter of the cellular events as they pass through the interrogation point. The larger the cell, the more forward scatter you'll have. Therefore, you should be able to see differences in that forward scatter versus side scatter plot when looking at yeast, which are inherently bigger than probiotic bacteria, to get that discrimination. I will say we don't get that request very often. We do see some products that contain both types of probiotics, but more typically we're either going to be looking at just probiotic yeast or just probiotic lactic acid bacteria or spores for that matter.
Great. Okay, move on to the next one, which is, are there complications when testing by flow cytometry when there are live cells? In this particular question, it was specifically bacillus cells, and also dead postbiotics are present. So are there issues with enumerating postbiotic in the presence of a live cell or vice versa?
Sure. There is potential for interference to occur when looking at a blended material containing a live spore and a postbiotic. In order to ensure that you can get an accurate postbiotic enumeration using flow cytometry in the presence of the spore, some preliminary testing will need to be performed in order to show that the spore material by itself will be able to be enumerated by flow cytometry and not interfere with the gate that is established for the postbiotic material, which of course is dead cells. For those that aren't familiar, postbiotic is animate cells and or their components that confer a health benefit to the host. And flow cytometry has shown to be a promising way to evaluate the potency of these materials, with of course a couple caveats, but because of course postbiotics obviously aren't going to be able to grow on a Petri plate, so it shows good promise for particular types of postbiotics. When you're blending it with a spore, the spore P-I positive or propionate iodide positive, or dead damaged spores, depending on the way you look at it, mustn't interfere with the postbiotic cellular population, as that'll end up skewing the results to an inaccurate level. But we have seen in some preliminary studies that we are able to discriminate postbiotics from the spore material. So then what you're able to end up do is evaluate the postbiotics by flow without the interference of the bacillus spore, and then proceed with a traditional cultural plate count enumeration or use the flow cytometry values for the spore enumeration.
Okay, since we're on the topic of postbiotics, let's stay there. And can you talk a little bit about the ability of flow cytometry to be used for enumeration of postbiotics, as well as what some of the challenges are in that particular area, since it is a growing field with increased interest lately?
Sure. Yeah, I agree. We're hearing more and more lately about postbiotic materials. I think we're seeing companies, invest in research to show that these sorts of materials do have health benefits associated with them. There is still work to be done in that area when it comes to forming a consensus definition. It is somewhat of an umbrella term, so it can mean everything from, an intact inactivated cell to a cellular component such as a protein or a short chain fatty acid, and it really comes down to what component is conferring the health benefit, and how that's being measured and how that's being applied in the clinical studies associated with these health claims. For flow cytometry to be used as a tool of enumeration for postbiotic materials, the cells still need to be somewhat intact. You'll see the term paraprobiotic used as an alternative to postbiotic for this particular subset of postbiotics, where the cell is still somewhat intact. It's not completely obliterated into its cellular components by whatever inactivation process is being used. And in those instances, then what we've found and what we have been measuring on a somewhat routine basis now for our client base who is interested in evaluating these sorts of materials, is that we can get an accurate enumeration expressed as a non-active fluorescing unit of the paraprobiotic or the intact postbiotic cell using flow cytometry. When the postbiotic is, as I mentioned, like completely obliterated into just its cellular material, it becomes more challenging, of course, because the measure of viability is based on cell membrane integrity. If you don't have any cells present and it's just a soup of cellular material, you may start to see inaccuracies in those values, and the correlation between what you had prior to the inactivation may not necessarily line up very well with what you end up seeing when you try to evaluate them by flow cytometry. The way that we measure postbiotics and the way that we're going to be demonstrating and providing information on a label to use as a dosage format for a particular health claim is still up for discussion. I think there's work that needs to be done in the industry to come to a consensus as to the best way forward. I think flow cytometry does have, as I mentioned, some benefits there, but only for a particular subset of postbiotics. It may be a case where the material needs a completely different type of measurement, whether that be like a HPLC measurement or something more chemical in nature, where we simply go back to a unit by weight, like another dietary ingredient. Traditional dietary ingredient will be measured like a vitamin or something like that, or something to that nature. I know that there's going to be ongoing discussions about the best way to evaluate these materials among the different industry groups, such as ISAP, and IPA also has a newly formed committee dedicated to looking at these materials and determining best ways for quality management, among many other things.
Interesting. Yeah, it seems like there's a lot more work to be done in that particular area as the products become more prevalent in the marketplace or get introduced into the marketplace in order to determine how we're going to measure postbiotics. But it also seems to depend on what's there or what the products are, which is really pretty interesting.
Yeah, and really, it comes down to understanding the mechanism of action with how it's providing that health benefit. Because the key portion of this, just like probiotics, the organism needs to confer a health benefit, and that needs to be demonstrated, by conducting a clinical trial. And so, what is the mechanism of action of this material for providing said health benefit? And sometimes that's not always known, and that's where it can get really complicated, because you may have a structure function claim associated with this material, but not necessarily understand the biochemical reactions that are taking place, or what portion or component of this material is indeed giving that health benefit to the consumer. And so better understanding of that aspect of it will really help drive the way that we're going to be measuring these for various dosage formats, in order to make sure that we're and companies are providing the efficacious dose in their products.
Okay, interesting stuff. Yeah, more to come there, I'm sure. All right, let's shift gears a little bit with some questions regarding the actual testing of live probiotics here, specifically, and I think this is in comparison to plate count. What is the limit of detection for flow cytometry versus plate count, which of course can be very low in the 10 CFU per gram or per milliliter, depending on the product being tested. So how do those two compare?
Sure. Yeah, as you mentioned, with plate counts, depending on the matrix, you can get a fairly low limit of detection, less than 10. If it's a liquid product, you could be looking at less than 1. Somewhat irrelevant for probiotic evaluations, because the efficacious doses of probiotics is much higher than that. But when you're talking about just a quantitative evaluation and the limit of detection associated with using Petri plates and a cultural enumeration, whether that be for a contaminant or a probiotic, being down at less than 10, flow cytometry is a bit higher. What we found in our initial validation work that we did a number of years ago, when we were validating our instrument and the method at our location, was that you can get down to 10 to the 5th, 10 to the 6th reliably, but it also kind of depends on the matrix and the concentration of the bacteria present. If you have certain material where the concentration of probiotics is significantly lower than the rest of the material present in the product, think like a fortified food or something like that, where you may be trying to measure probiotic cells in the millions in the presence of a lot of other extracellular material. It becomes increasingly more difficult to mitigate the debris interference that you can see. And really, in order to get the best evaluation by flow cytometry, you're going to want to have enough dilutions in your assay to have a nice clean sample to run on the instrument. If you're not able to dilute out far enough to have that clean sample, then you're going to run into issues with debris and interference in those sorts of situations. So it works really well with raw materials when we're talking about hundreds of billions of cells per gram. Pre-blends, dietary supplements typically have dosage formats that are high enough that we can dilute to get to a nice clean sample in order to run by flow cytometry. But when you start talking about the more complex matrices with significantly lower concentrations of probiotic bacteria, it becomes increasingly more difficult. And there are ways we can mitigate that. You kind of have to take it on a case-by-case basis. So, for example, we could simply run we could simply dilute out farther and then run more volume through the instrument in order to get to an event count that we can use to accurately estimate the number of probiotic bacteria present, because you do need to have a certain threshold of a number of events, similar to colony counts, where we're looking at a range of 25 to 250 or 30 to 300 colonies per plate in order to make a good statistical estimation of the potency of the product. Similar vein in flow cytometry, just with different numbers. We really want to see event counts that are above 1,000 in order to get those sorts of accurate representations. If we're looking at event counts that are just 100 or 200 in the gate that we typically are using for live cells, then obviously that accuracy or that the inaccuracies increase, as well as the repeatability. Other ways that we've looked at to mitigate debris include things like differential centrifugation. It really comes down to the type of preparation you're doing. So various filtering steps, using traditional centrifugation in order to separate out the debris from the probiotic cells. But like I said, it really does depend on the matrix, so it needs to be taken on a case-by-case basis. But reliably, 10 to the 6th, 10 to the 7th concentration of probiotics per gram of material, and you're going to be able to get an accurate and repeatable value by flow.
Okay, good. So I'm glad you came back to it at the end. So the 10 to the 5th, 10 to the 6th, even 10 to the 7th seems to be where you'd want to be on a per gram basis in terms of being able to reliably get an information value, right? But there are some matrix dependent issues there, which is kind of leads us into our next question, which is what type of matrices or finished products have you tested on flow cytometry, and what are some of the challenges associated with those perhaps more complicated matrices?
Sure, yeah. We've been a contract lab, we've seen all sorts of different matrices that have been evaluated or attempted to be evaluated by flow cytometry. Microencapsulated bacteria is one that has its own unique set of challenges associated with breaking down that microencapsulation. And that can also be kind of tied into the various matrices that we can see these probiotic bacteria being introduced in. So whether that be, gummy or a fortified beverage, various dairy products. Again, it goes back to the same principle that you need the concentration to be high enough where we can dilute out to get a clean sample that doesn't have that interference that we're speaking of. But we've tried all sorts of things. We've tried granola bars, with a yogurt coating on top of it. I could say those are particularly challenging. But if you could think of it, we've probably at least tried it by flow cytometry. A whole host of different dietary supplement finished products with typically good success with those. There are certain instances, depending on the other active ingredients present in the dietary supplement, when you're talking about a finished format, where you could see some interference from other things fluorescing or taking up the dyes if there's other organic material present. But typically, those dosage formats are in the billions of cells per dose or per gram, and in those cases, we are able to get nice precise counts using flow cytometry.
Okay. All right. Well, let's talk a little bit more about plate counts versus flow cytometry. So, what do you see in terms of the differences you'd encounter on the same product for a CFU versus AFU? And then further, how would you see those differences change over the course of a shelf life of a specific product?
Sure. Yeah, so taking the same material, let's take, for example, a standardized raw material that's 100 billion CFU per gram of lactic acid bacteria. Choose your lactobacillus or bifid. When evaluating these materials by plate count compared to flow, what we found in our initial validations that we performed, and what we've seen, as in our experiences running these on a almost daily basis, is the precision of the flow cytometry evaluation is typically better than plate counts. What we found in our initial validation was that relative standard deviations for these sorts of raw materials was approximately half that of plate counts. Again, those numbers will depend on the matrix being tested. So this is, I'm talking specifically for the raw material, strains that we test on a regular basis. They are different units of measure, and that's an important aspect of flow cytometry testing compared to plate counts. Plate counts is a colony forming unit. It's the cell's ability to replicate. Flow cytometry is active fluorescing unit. That's based on cell membrane integrity. They are not interchangeable. Usually when a culture, a fresh culture, let's say, recently produced, not under any sort of stress, those values, CFU and AFU, will be very comparable. If there's stressors that are added to the material, or we're just looking at the stability of the raw material over time as it slowly degrades, that's when we can potentially see differences in the values of the results by flow cytometry and the AFU compared to CFU. And what we often find, especially if we're looking at stress conditions or some sort of accelerated stability, and there's published literature out there on these sorts of evaluations, is that flow cytometry tends to have a higher count as the product ages in these accelerated stability conditions. And so what you see is that divergence as the plate count lowers. The flow cytometry might be lowering some as well, but not nearly as much, where the plate count may drop off a cliff at one point. You're still seeing the AFU measure remain relatively higher. And there are a few theories on why this is happening. Of course, it's a different unit of measure, but also there's the idea that a lot of these cells become in a state where they are viable based on their membrane integrity and their ability to uptake the dyes associated with the protocol that we're running for flow cytometry, but not culturable, meaning even though they're by definition alive, and a probiotic by definition needs to be alive, although it does not say how it needs to be alive per se, that it can't replicate on a plate. And therefore you have these viable but not culturable populations of cells that can increase over time depending on the type of material, the type of strain. All of these sorts of evaluations are very dependent on the strain itself, as well as the matrix. So if you start looking at incorporating different strains in a blend with other ingredients, a finished format, that correlation or lack thereof is going to differ over time. And so while I would love there to be a universal correlation between flow cytometry and plate count evaluations, it just isn't the case because of what I just described. And you really need to take it on a product by product or strain by strain basis, and generate a lot of data to support any sort of comparison that can be used in the future. And then even then, looking at different lots of materials and how consistently a particular strain is produced, I think it would just need to be continually evaluated by both to ensure that what you're representing based on the efficacious dose, because of course it needs a health claim, and what you're putting on your label can be justified scientifically. So having data to support that is going to be critical, and it's not as easy as saying, apply this equation and you can take your AFU count and turn it into a CFU. And that's one of the big limitations with flow cytometry, and why we haven't seen it, take off as much as you would think, based on all the other advantages we've seen. When you talk about increased precision, you're talking about less overages, and who doesn't want less overages in their product, because you have to take into account the variability of the plate count results as well as the degradation, but it's not a CFU. And if you're going to be using flow cytometry, for a claim or for a label, then it needs to be substantiated in some way to however that health claim was established, and there needs to be data to support that.
Okay, I'm glad you kind of summarized it there towards the end, that each product really needs to be treated differently in terms of the strains that are present, and then the matrix itself when you're comparing the AFU to CFU to really establish what those differences are, and that there's not one size fits all rule. So let's kind of stay in that same area on our next question here, which is when you report flow cytometry, what are you reporting? Is it just the live? Are you reporting the dead and injured as well? And then again, how does that compare to the colony forming unit or the plate test, and what are the advantages and disadvantages there?
Yeah, so typical report, and we run flow cytometry potency evaluations on a routine basis through our routine channels in our laboratory. We will give a live, dead, and injured quantification of the probiotic population of that sample. When we're evaluating spores, it's a little bit different because we don't have as good a separation between the dead and injured cells. So what we end up reporting is a live spore count and a dead/damaged spore count. And that's just based on, again, the same principles of membrane integrity. And in similar respects to what I just mentioned in regards to plate count evaluations, it's going to need to be taken on a case-by-case basis to look at potential differences between the two and why that may be occurring. Again, if we're looking at fresh product under not a lot of stress, those values will typically be very similar to each other. But yeah, it really does depend on the organism and the matrix that's present in.
Sure. I really like to think about having the dead and injured cells as a big bonus in terms of the additional information you're getting there, especially if you're doing an investigation or something like that. You can really see more of what's going on with that product in comparison to just a CFU count, which is just what's it live and able to grow on the plate. So there are, in my mind, there's always some additional value there.
Yeah, that's a good point. That's a good point you brought up, because it can be used as an investigatory tool. We do this on a regular basis as well. We use it for a variety of reasons. Clients will use it for release testing. Other times, a client may have a low plate count results. Let's just take an example of a finished product capsule. It's expected to be 10 billion AFU per dose. Or CFU per dose, excuse me. They're measuring their efficacy by colony forming units. We test it with the plate count evaluation. We got, say, half that, or maybe 1/10, a log lower. So we have 1 billion CFU per dose. Why is that the case? We can apply flow cytometry as a tool to help investigate potential root cause for the out of specification result. So say, for example, we test it by flow cytometry and we see, yes, we have the 1 billion live cells, but we also are seeing now we have 9 billion dead cells or more. And there's actually a much larger population dead cells present for whatever reason. Maybe it's an old sample under stability, or temperature abuse during shipping, or pick your reason why you may see that. But flow cytometry allows us to see that, where plate count we wouldn't be able to obtain that information just based on what's able to grow or lack thereof on a plate. Another instance would be, it's testing by flow cytometry at 1 billion CFU per gram, but we have even lower injured and dead counts. And so that tells me, when I'm looking at these sorts of investigations and trying to root cause, well, perhaps it was never there in the first place, and was there, a formulation error or something upstream that maybe had caused the product to not contain the set amount of bacteria or probiotics that they were expecting. So it can be really helpful in those respects. It's not going to necessarily give you a perfect answer every time, but we've used it for those sorts of investigations with success in the past, to help inform the clients, of ways to mitigate those sorts of results from happening in the future, or help explain, okay, well, what's really going on here, and it can be a helpful tool for that part of the quality management system.
Sure. So not only for release, but also for investigation can be a tool as well. So that's interesting. Okay, let's move on to what I think is probably the biggest and probably most important question on this list, which is how do regulatory bodies treat AFU claims versus CFU claims? And are there companies that are in the marketplace today using AFU for release and labeling their probiotic products? Because that's kind of where we'd like to get this technology to, I think, in many of the people's minds who might be attending this call.
Sure. Yeah, that's a good question, and I'll speak for regulations in the United States. They vary depending on the country. So what we see here may be different than what Health Canada is prescribing, or the EU. So that needs to be my answer needs to be prefaced with that. We'll be looking at just the United States for this, since that's where we're based. But the way the FDA looks at these, and with dietary supplements specifically, is that the dietary ingredient by regulation needs to be measured in milligrams. And for those in the industry that have been working with probiotics, know that doesn't actually inform the consumer about the actual potency of the probiotics present. It may have been used in a clinical trial, and so in that way it could potentially support it, but 100 milligrams of a certain organism isn't going to be the same number of CFUs than a different one, and so it really ends up being somewhat misleading. They did put out, the FDA did put out a guidance document in 2018 stating that along with milligrams, companies could also put CFU or colony forming units as another way to label the potency of probiotics present. And of course, that's evaluated by plate counts. That's considered the quote unquote gold standard of enumeration, and that's what's typically used when you pick up a dietary supplement off the shelf, and you're looking for what what dose is present. They do in that guidance document allude to the fact that there are other technologies out there to measure these potencies. Flow cytometry, real-time PCR or digital PCR is another technology out there that has shown some promise as an alternative to plate counts. They recognize they exist, and essentially what needs to happen is if you want to use flow cytometry for a label claim, it needs to have evidence to substantiate the health claim associated with it, and there needs to be data to support that whatever is on that label will support, or is the amount required that has been shown to be efficacious for the health claim associated with the probiotic. I think if the FDA, came and questioned, as long as that information was available and it's scientifically valid, that there is no reason to say that you couldn't put an AFU on a label. It just needs to have that substantiation. And whether that means it's a correlation to colony forming units, or it's, the health claim was measured in milligrams, or it has lack of a measurement. I mean, there's the notion that, all clinical trials were done in CFU, and so it's going to be impossible to use flow cytometry because of AFU, and there's no correlation. But actually, it's not entirely accurate, just because there are a lot of clinical studies out there that used a different unit of measure altogether, or didn't establish or didn't state explicitly the dosage format in CFU, for example. And so that argument kind of becomes a little bit muddied. But really, it comes down to substantiation. And I don't want to speak for the FDA. I obviously don't work for the FDA, but as a scientist, understanding the what's required to show efficacy, and as long as there's data to support that, I think they're understanding, and that's supported in that guidance document.
So my takeaway there is, yes, it could potentially be used for, at least for labeling, but there are caveats, and you got to be able to be sure that you're substantiating. That sum it up pretty well?
Yes. Yep.
All right. So let's move on to one kind of future-looking question. At least what I think is a future-looking question. Are you able to differentiate species of probiotics in a blend by flow cytometry?
Good question. Yeah, I agree. That is a forward-looking question. There's current state. What we're able to evaluate is total live, dead, and injured. We have shown the ability to differentiate certain spore materials, specifically Bacillus subtilis and Bacillus coagulans in a blend, based on their different affinities to uptake the dyes associated with the protocol that we're running. When we start talking about evaluating different species or strains in a blend by flow cytometry, we need to start applying different practices in order to separate those species and be able to differentiate them based on the various emissions and the readings that can be associated with them. So one way that's been shown to work in a limited fashion, and there's some publications out there that show that using polyclonal antibodies and tagging different strains does have the ability to differentiate different species and strains in a blended material using flow cytometry. There's also in situ fluorescent hybridization that has been is being looked at in conjunction with flow cytometry. That is a different way to evaluate and differentiate different species on a limited basis. And the key to that aspect is you're going to get to the species level and not necessarily the strain level. And so when it's a strain specific health claim, then you have the caveat of, well, if you have two different strains of the same species in a blend, it's going to be that much more difficult to differentiate the two. I mean, by play count, it's impossible, for that matter. I mean, in a similar vein, play count has the same sort of limitations. There's a few assays here and there that can determine the species in the presence of a blend. Think like Lactobacillus acidophilus has an ISO method associated with its selective enumeration. Fairly straightforward to get to the genus if you're looking at bifidobacteria compared to lactobacilli or the genus formerly known as lactobacilli. But differentiating strains is nearly impossible by play count. Very difficult by flow cytometry, and even other technologies. When you start talking about quantitative PCR evaluations, it becomes increasingly more difficult when you have two different strains of the same species and trying to find genetic differences between those two in order to develop, the primers and probes required to get differentiation and differential quantification by a PCR reaction. It poses a lot of challenges, and it beckons to, it raises the question as to, when you're formulating a product, if you need species or strain level quantification in a blend, are you going to pick the species that are going to be more easily differentiated by the tools that we have? Does it make more sense in that case? And that may depend on, where you're selling the product to and the regulations required in a particular country to show that you have X number of, this strain versus this strain in a blend, or if it can just be based on, the input level, which is a common way to do it. But yeah, there's promise with flow cytometry specifically, going back to that, that there will be various applications that will be able to differentiate to the species level, but we're not there yet from a production lab or standpoint, or a third-party contract lab standpoint.
Sure, interesting. Okay, well, I have one final personal question that I wanted to ask you, and then we'll move on to just making sure we've covered everybody's questions that have come in throughout the webinar. So I just wanted to ask you to do a little bit of prognostication about where you see this technology going. We flash forward a year or 5 years. Where's flow cytometry going to be in the marketplace in terms of a testing methodology and in comparison to plate count, or just as a standalone? This is really your opinion.
Yeah, in my opinion, yeah. I think eventually, and I don't think it's going to be a short-term thing. I don't think it's going to be next year. I don't think probably not even 5 years, but I think there's enough advantages with whether you're talking flow cytometry or digital PCR or something else perhaps that hasn't even been looked at yet, that eventually there's going to be a paradigm shift with the way that we're evaluating these materials, moving away from the plate count, and the traditional kind of, it's always been this way, it's the gold standard. We could have run this assay 100 years ago and got the same number that we're getting today, to something more sophisticated, where we are able to differentiate strains in a blend, where we can get more precise results, where we can get real-time data to help, drive decision making in a faster paced environment. There's obviously, I touched on a number of hurdles associated with that paradigm shift and making it happen. But in my opinion, I think it's only a matter of time. How much time is hard to say. We could be talking 10, 20 years for all I know, before we see something like that. But as you see the technologies advance and become more and more sophisticated, whether we're talking flow cytometry or we're talking genetics or genomics applications associated with these sorts of evaluations, that it is only a matter of time.
I would tend to agree with you there, so thanks. And in looking at the questions that came in throughout the webinar, it looks like we only have one that we did not already address, and that's specifically how long does it take to process a sample and get final results, and perhaps in comparison to a plate count there as well.
Sure, yeah, that's a good one, because as I mentioned, it's real-time data, but of course there's preparatory steps involved. So when we're talking hours, a single sample using the ISO protocol we typically use, from the time you weigh it to putting it on the instrument, you're looking at about an hour. And then instrument time, we typically perform 6 instrument runs per sample, a couple different dilutions. So you're looking at about a half-hour instrument time. So for a single sample, from the time that we weigh it to the time we obtain results, you're looking at maybe a couple hours at least in order to get that result. So that's, huge advantage. Plate counts, there's also that preparation step. Sometimes it's a little bit shorter depending on the assay. but then you have plates that need to sit in the incubator for days in order to get that estimated value. And so we've found certain applications where our results needed really quickly, and so flow cytometry has come in handy. Obviously, it's been used for a long time on the side of fermentation optimization, and the manufacturers who are producing the strains have been using flow cytometry for years now to do just that, because they need quick answers on the viability of the cells when they're optimizing fermentation processes. So yeah, a couple of real distinct advantages there.
Sure. Okay. Well, that's certainly an advantage to plate count there. So thanks. All right. That covers all of the questions that we had for today. So just wanted to say thank you to everyone who attended. I hope you found it valuable and informative. I know I did. So thanks again, and have a good rest of your day.
Thank
Thanks,
you so much. And just as a reminder, we will be forwarding a copy of the recording within 3 business days. So if you missed any of those, but thanks again, Anjay and Dave, and have a great rest of your day.
You bet. Thanks, everybody.
Thank you.